All right, I'm gonna run through what you need to do for the pipe cleaner lab. You're gonna need 2 sets of pipe cleaners and a pair of scissors, preferably the old pair because you're gonna be cutting the wires and we don't wanna ruin our good ones. So the first thing you're gonna do is we're going to cut these pipe cleaners into our chromosomes. We're gonna make 2 cuts so that we end up with 3 pieces of each pipe cleaner, 1 small, 1 medium, and 1 large. And you wanna make sure that you can easily tell them apart. So it's kind of easier to cut these one at a time. So here's all of my small chromosomes. Okay, then I'm gonna cut the medium ones and the large. Okay, so when we start, we're gonna just start with our diploid cell that has 1 copy, of each one, like this. Okay, so we have 6 total chromosomes here. Then during S phase of interphase, these are going to replicate. So I'm gonna show that now by putting on the matching pipe cleaner, which now these have become sister chromatids. Each of these are an exact copy of each other. This is still a diploid cell. It still has 6 chromosomes, but each of the chromosomes just has a copy. So then we're gonna go through mitosis. The front page of the pipe cleaner lab is mitosis. Remember that the purpose of this is to replicate the cell. We're gonna start with that diploid parent cell we saw, and we're gonna end with the 2 diploid daughter cells. So first we're gonna do prophase. Prophase is where all of the nuclear membrane is gonna dissolve, the spindle fibers are gonna start to move, and centrioles move to the poles, and the chromosomes are gonna condense into these visible shapes by supercoiling. Okay, then metaphase is gonna occur, and they're all gonna line up along the equator of the cell, just like this. We'll just line them all up. Okay, so notice that in mitosis, they just line up by sister chromatid. They don't form those bivalents where they associate with their other homologue. We just have 1 row, so that in anaphase, we see separation of the sister chromatids to each side as they're pulled by spindle fibers, like this. Okay, and then eventually we get to telophase, where the chromosomes are gonna decondense, so that's kind of hard to model because I don't really know how to decondense a pipe cleaner. The nuclear membrane reforms, and now we have 2 cells that are identical to what we started with, each with 6 chromosomes. That's mitosis. So now let's contrast that with meiosis, which is on the back of the pipe cleaner lab. So we're gonna start with the same thing, okay? 1 cell that's got 6 chromosomes, right? Each of these, one's from mom, one's from dad. So you have 1 copy from each of the parents. So again, the first step is exactly the same, where all of these are going to be duplicated in S phase of interphase. So now we have sister chromatids again. Okay, but what's special is that in prophase 1, okay, in prophase 1, these bivalents are gonna form through synapsis. So we're gonna have a set of sister chromatids that's gonna line up right next to its same size set of sister chromatids. So the one from mom and the one from dad. Remember, these have the same genes at the same locations, but they might be different forms of the genes called alleles. So what's special that happens at prophase 1, I'm gonna use this one as the model, is that crossing over can occur. And when that occurs, there can be an exchange of genetic information. Now I'm gonna show that by cutting a piece of the end of one of the chromosomes, whoops, like so. That's touching this one. And you're welcome to twist these together if you'd like. I'm just crossing them. But I'm gonna twist this to show you that they're exchanging genetic information. So now there's just a little piece of that chromosome there and a piece here. Okay, so that's an example of crossing over that's occurred here during prophase. Okay, and you could do it in all of them if you wanted to. Then we're gonna get to metaphase. All of these are gonna meet in the middle. They're gonna line up like this. Notice this is very different from mitosis, where they all just lined up in one row, and so the sister chromatids could separate, because remember that in meiosis 1, we have separation of these two sets of sister chromatids, this homologous pair. So then anaphase, we're gonna see separation like this. Okay, and then telophase, it's gonna decondense. Now we have 2 cells, but remember that meiosis has 2 divisions. So then we're gonna show the second division. Prophase, let's go into metaphase. Now I'm gonna show these, hopefully I'm gonna move those over. These are happening now in separate cells, so there's 2 different cells here, pretend. Okay, then anaphase, where we see separation now of the sister chromatids, like such. Okay, and so our final product at the end is going to be 4 cells, each with half of the genetic information. And notice that they're different, crossing over has occurred. Oh, I didn't really mention random orientation, which I should have, which was earlier in metaphase, that there was a 50-50 chance of which side during metaphase that each of these lined up on, okay? And that occurs both in metaphase 1 and metaphase 2, random orientation. So maybe the one from dad goes this way, maybe the one from dad for this one goes that way, right? So depending on which side of this plate these line up on, right, it's gonna split in very different ways. So you're gonna have different gametes every single time. Okay, hope that was helpful.